Hello everyone, welcome to my channel. This is the second video that I use English to explain about the syllabus of STPM Biology subject Biology semester 3. This year is 2021. I think nowadays many school already begin their sem 3 lesson lah. But I think that only touch about the chapter 1 for about the intro only. So you guys that begin the sem 3 that you will also can watch this video, because this video I will talk about the sem 3 syllabus chapter by chapter. I will focus about the chapter that you are need to focus and what will you learn in that chapter. Okay, first, the sem 3 biology, the theme we have to learn is about the ecology and genetic. Yes, there are two themes that you have to learn, and these two themes are totally different. Ecology we have 3 chapters and genetic we have 3 chapters. Okay, for chapter 1, it is the taxonomy and biodiversity. Chapter 2 is ecology. Chapter 3 is selection and speciation. While in chapter 4 is the genetic topic. It is the inheritance and genetic control. Chapter 5 is gene technology, and chapter 6 is biotechnology. So altogether, since we have to learn 6 chapters. And before I enter the briefing syllabus, I just want a gentle reminder that sem 3 biology is the most difficult sem in STPM biology. So, and another thing is sem 3 do not have repeat paper, only have one chance only. So you have to take more effort compared to sem 1 and sem 2. Okay, we begin to chapter 1. Chapter 1 is about the taxonomy and biodiversity. So firstly, we have to know that what is taxonomy? You have to know what is taxonomy and their importance and its importance. Taxonomy, why they are important. So if you know that generally you know that in our world, in Earth, the world, there are more than billion and billion types of species, right? There are many species, maybe more than 100 billion atau 100 billion. There are many types of species in the world. But how do we naming the species and classify the species? This is why we learn the taxonomy. So one of the importance of taxonomy is we have to classify the species and name the species. And so the second one, you will learn about the concept of species. What is species? How do we define the organism is known as species? Dia ada peraturan dia. It has the rules and condition there. We cannot simply say that the new organism it is not a species. No. We have the rules and regulation to define that the organism is species. And the third one is taxonomy hierarchy. This is the classification of the organism. You have to know. Actually, we have 2 types. It is natural classification and artificial classification. So you have to know that there are difference, and also the generally classification we use is artificial classification. Artificial classification is the human-made one, not the natural. Natural is the organism have. Artificial is the human make an observation and make one conclusion that. So in human we have already create a 7 level taxon. Taxon you have later you will learn. The largest one is kingdom. Then you will enter phylum. Then you will enter class, order, family, genus, and finally is species. This means that if you want to define any species, actually you have to know this species of genus, their family, order, class, phylum, and kingdom. Yes. So if have a new species, you have to know which kingdom it is from. You need to know. So what is kingdom? Kingdom actually we have only 5 kingdom. We have only learned 5 kingdom in STPM. 5 kingdom. Another kingdom is known as prokaryote. This kingdom actually is prokaryote. Prokaryote actually you have already learned in SEM 1. So SEM 3 you will not learn already. So in SEM 3 you will only learn about this 4 kingdom system. And each kingdom system has many types of phylums. And one phylum has many types of class. And overall as well as like that until species. But in STPM, you don't need to worry that. At STPM, you only learn detail in kingdom, phylum, and class only. Order, family, genus, we will not learn too detail. You can find the information, but in STPM syllabus, you don't need to learn too detail about this. Okay, but kingdom, phylum, and class you must learn. Especially for kingdom and phylum. And kingdom, we have 4 kingdom you have to learn: Protista, fungi, plantae, and animalia. Okay, so in Protista and fungi are more easier because you only learn one phylum only. But for Protista, it is special. It has 2 sub-kingdom, that is Alga and Protozoa. It is unique. Other kingdom system that don't have sub-kingdom, but in Protista it has sub-kingdom. And sub-kingdom for Alga and Protozoa, there are many types of phylum, but in STPM syllabus we don't need to learn all the phylum. For Alga we only learn about Chlorophyta, and Protozoa we learn only about Jusmatigana only. And what will you learn in this topic? You will learn about the characteristic. You need to know the characteristic of all level. It is characteristic of Protista. What is its characteristic? And what is the characteristic for Alga and Protozoa? And what is then you have phylum. What is the characteristic of Chlorophyta and Jusmatigana? And also the second one, you will learn about the example of species. Which species is beyond on the Chlorophyta and Jusmatigana? You have to know. I think Chlorophyta has 2 example one to learn, and Jusmatigana has 3 example one to learn. All the species you have to know the cell structure and also their function, like that. You have to learn that. Okay, then about the fungi. Fungi is the easiest because it has no sub-kingdom and you have only to learn one phylum only. Only one phylum you have to learn. That is Zygomygota. And the species also learn easy. Mucor species. You only learn mucus. Example of the Zygomygota species is mucor species only. Fungi is the easiest. And also you will learn about the characteristic of fungi, characteristic of Zygomygota, and the cell characteristic of mucus species. Okay, this is easier lah. Protista and fungi are more easier because not too much thing. The difficult part is the plantae and animalia. Plantae actually is plant, animalia is animal. Just in taxonomy terms, we cannot say plant and animal. We have to say plantae and animalia. For plantae, there are 4 phylum you have to learn. 4 phylum. And animalia, you have 6 phylum you have to learn in STPM. Actually, for animalia, there are more than 10 phylum. If you actually have get research, there are more than 10. But in STPM, you only learn 6 phylum only. But although only 6, but it also too much. It also much. Okay, for plantae phylum, one of the phylum called Angiospermophyta, I think. One of the phylum. This is one of the phylum of Plantae. You will learn about the class. There are two class you have to. There are two class. Another level, the third level, you have to know. Two class. You have two classes you have to know. This means that you in this phylum, you already want to learn characteristic of Plantae. Then each phylum you have to know the characteristic and also the example of the species of that phylum. But for the phylum Angiospermophyta, you want to learn about the characteristic of Angiospermophyta and also the characteristic of these two classes. These two classes, you also want to learn about the characteristic and also the example of beyond to these two classes. What are the example species beyond to these two classes? You have to know. Same that six for the Animalia. Animalia have two phylums that you have to learn until class. That is Arthropoda and another is called Chordata. These two phylum, you also need to learn until class. The class are not little like the Angiospermophyta. Arthropoda, I think, has 6 classes. If I am not mistaken, there are total 6 classes you have to know. And for Chordata, there are total also 6 classes. If I am not mistaken, you can refer to your textbook. There are total 6 classes. Each classes you have to know. This means that 6 phylum, you have to know all of the 6 phylum characteristics. And for Arthropoda and Chordata, you not only know the characteristic of Arthropoda and Chordata, you have to also want to know the characteristic of each classes. Each classes you have to know. And the example species of each classes you have to know. Why? Because it is under the syllabus MPM. MPM syllabus ada, so you have to learn. And other 4 phylums in animal, you only learn until phylum only. Class you don't need to learn. But if you want to learn, you can also make a research in your internet or others international reference book also can. But the exam will not come out because it is not in the syllabus. But for Arthropoda and Chordata, it is under the syllabus until classes. So this is more difficult part in this chapter because understanding is easier, but there are many terms that you have to memorize, especially the example of the species. Example of the species are not easier to memorize, but you have to memorize also. Okay, these are the for this chapter. So this is the importance. The most important part in this chapter, so you have to make more comfort in this chapter and advise you to take note, to make a note in this chapter, especially for differences. Maybe for phylum and phylum, differences between phylum and phylum. You may do a table, then you make a differences. Okay, then about the taxonomy chapter is finished. Then we will enter the biodiversity topic. Okay, you have to know that biodiversity in Malaysia. So firstly you have to know the class level of biodiversity. There are actually biodiversity we have 3 class level, and each class level you have to know their definition, also their application. This means that you not only to memorize the definition only. If you memorize without understanding, it is tak ada guna, benefit to you. Why? Because normally in object question, it will come in object question. It will give you a class level name and also description. One of the description, any description it can give. So if you want to, based on that description, match the correct class level, you have to. So it is more challenging if you do not understand. Just memorize a definition. You cannot answer that kind of question. So you have to understanding what are the definition actually stated. Another topic is importance of biodiversity. This one just memorize saja lah. Memorize only, not too much. Okay. Then the 14.4 is threaten to biodiversity. Ancaman kepada biodiversity. Apa ancaman dia? So first one you will say factor that threaten about the biodiversity. Apa faktor menyebabkan biodiversity diancam? Ancaman lah, apa yang mengancam biodiversity di negara kita. You have to learn, and another is you have to also learn the step caranya untuk menyelesaikan ancaman ini. This kind of question actually already have come out in essay question 2 times, I think 2 times already, and each time carry 10 marks. It is very high, but this kind of question, it looks easy, but not easier to score, because the reason like macam pengajian am pun boleh keluar macam seolah macam ni kan. Tapi awak pun tahu lah pengajian am isi dia macam mana. Saya pun tak tahu cakap banyak. Awak sendiri pun tahu ada ambil pengajian am. So same as biology, you cannot give the too general, too normal answer. Like the step effort, you say that take a legal action like that, or you say that improve the law like that lah. Macam too general. You cannot give too general answer. You have to give the biological technique answer. So this 2 question, maybe you have to find more about the information from the internet. Okay, these are my advice to answering these 2 kind of question. And the last topic of this chapter is conservation of biodiversity. Pemulihan biodiversity. There are 2 types of conservation. There is in situ conservation and ex situ conservation. And this conservation actually maybe you have also want to find more information from the internet, because textbook, you see the Oxford textbook, the information is too little, and got 1 year exam essay it's come out importance. I don't know which conservation already, in situ or ex situ, 10 marks. I know 10 marks, but I already forget which part. It give 10 marks. But if you see the reference Oxford textbook, you cannot get 10 marks if you just read the Oxford's book. So if you want to get higher marks, if that kind of question come out, maybe you have to see more, see the reference from the internet or journal, other journal article, or other ecology textbook. If you want to refer Campbell also do not have this conservation. Not too much. So if you want to refer, you may refer to the ecology textbook. Okay, so this is all about the chapter 1. In conclusion, chapter 1 is easy, not too difficult. Just there are many things you have to memorize only, but understanding is easier. Understanding is easier, then you have to take time to make a memorization. Next chapter, chapter 2, is chapter ecology. This chapter is, the name of the chapter is same as the theme of this semester. Okay, in this chapter, there are total 6 subtopics you have to learn. The first subtopic is about the level of ecological organization. If you learn in chapter 1, taxonomy also have the level, right? There are 7 levels. So in ecology also we have the level. It is hierarchy in ecology. There are many levels in ecology also. You have to know that. And then the second one is interaction biotic and abiotic factor. This one actually you have already learned in SPM. Apply to what is the biotic and abiotic factors, you have to know. And the third one is type of biomer. Okay, biomer actually is one of the level in ecology. It is the second largest level in ecology. And there are total, I think, have 8 types. They maybe 8 types. I already forget 8 types of biomer you have to learn. If you don't know that, I give an example like a tropical rainforest. Tropical rainforest, Malaysia, right? Malaysia is one of the area that is tropical rainforest is one of the biomer, and another one is desert. Desert is also another one biomer also. There are 2 types already. There are total and 8 types. If I am not mistaken, you may refer to the books. And the last topic is nature. You have to know that what is nature, and also there are many types of nature you have to know. Okay, then the second topic is about the biogeochemical cycle. Okay, you will see that bio, this means that there are the biology, it has include geography and also chemistry. So these are the 3 components that it combined together, so it called biogeochemical, and it is the cycle. It is the cycle. Actually, this you have learned. That is nitrogen cycle in SPM, right? Nitrogen cycle. But STPM you not learn already because it is at the SPM syllabus. STPM you are luck not learn. So you can forget it at all. Okay, you just learn these 3 cycles: carbon cycle, phosphorus cycle, and sulfur cycle. In this cycle, you have to learn what is the first one is the flow chart. The flow of the cycle, you have to know. And the second one is the organism. And the role of the organism, there are some organisms play a role in the cycle. You have to know what organism that play a role in this cycle. You have to know that. The examination normally it will come up in objective question. It will give you like that. Like that. There are many flow chart like that. And there are many arrows like this, like this, like this, like this, then like this, like that lah. You have many arrow one, and it will give you something name naming like CO2, CO, sediment, macam have many names, and it will give you ABCD. It will ask you which statement is true about from this diagram, or it will give you like a S. What is S? Like that. Or which statement description of X is true or is false, maybe like that. BJ question lah. So this one you have to know detail about the flow of the cycle. You have to know detail. If you don't know detail about the flow of cycle, then this kind of question you will not understand. You will not understand what is the arrow means. If actually the arrow has the mean, it has the mean one, not the arrow plot syok saja, not syok. Ada maksud dia. So you have to know. Okay, this is the second topic. Okay, the third topic is the energy flow. Actually, you also will learn in SPM also. That is, you will know that primary consumer, secondary consumer, tertiary consumer, right? Primary producer also, and the higher consumer, when they obtain energy from lower consumer, only have maximum 10% of energy will be obtained, right? This one also will come out in STPM also. This is there where you will learn. And also you will learn about the intro first. That is the new thing is law of the thermodynamics. There are 2 laws, first law and second law of the thermodynamics. And also you will learn food chain and food web. Okay. And the second topic is the energy flow in tropical forest and lake. Okay, you have to describe the energy flow that occur in tropical forest and lake. You have to describe that, and it will maybe come out in exam question. It maybe come out with describe the energy flow in tropical forests, like that. Maybe 10 marks, like that. But until now, no passing question not come out yet. But in old syllabus question, they are come out this kind of question. But new syllabus not yet. So you have to maybe you have to make a comfort with this. But it's actually easy. Just describe only if you already know the concept of the energy flow, then you will easier to write out the description easily. And the fourth topic is the population ecology. This is the most popular topic in this chapter. Many times structure question and essay questions banyak keluar banyak kali lah. It come out very how to say keluar lah. Like the exponential curve and logistic curve already come out 2 times in structure question. Survival sheet and KR strategy banyak lah. Survival sheet structure question have come out. Essay question pun come out. And KR strategy essay question come out 2 times. Already come out 2 times already. So these are the topics most popular lah in this chapter. Banyak keluar. Lalu in the first you have to know the population. You have to know the population growth curve. Populasi dia bentuk graph dia. There are 2 types of curve: the exponential curve and logistic curve. You have to know the shape of the curve and the description of the curve. Why that population provide this kind of curve, and what is the differences between these 2 curves? There are differences. You have to explain also what is the mean. This curve provide what meaning to you. Okay. And another one, and this is the graph. And third one is survivorship. Survivorship is macam lifespan, because in all organisms in the world, not all organisms have the same survivorship among their life. There are some organisms that have different survivorship. So there are total 3 types of survivorship you have to know. Each type also description, you have to know their description and also example. Which organism beyond that type of survivorship? I already say this question, even nominal structure question or essay question came out many times. Structure I think 1 times, essay have 2 times already. Popular question, one of the popular question. And the last one is K and R strategies. What is K strategy and R strategy? You have to know their difference and then make sure you write correctly. There is K and R. K must be capital letter, R must be small letter. If you write K in small letter, sorry, wrong. Even your answer is correct, sorry, wrong. If you write in capital letter, sorry, also wrong. This is bio. Bio spelling is very strict. There is no other argument that examiner will give you marks because your spelling is already wrong. And this question already come out in 2 years. 2 years come out in essay question. And also you have to know the example. What are the example of the organism that belong to K and R strategies? You have to know that, and the differences. Mainly essay question will come up with differences between K and R strategies. Okay, next one is carrying capacity. This also the most popular question for definition. Define carrying capacity. Already come out in 2 times in structural question. What is carrying capacity? You have to know the definition, and also then you will learn about the sustainable development and also the factor limit the population size. Apa faktor yang menyebabkan populasi size tidak akan menjadi tidak akan menjadi sampai dah hujung lah. It will not increase until infinity. No, population size cannot increase until infinity. Why? There are the factor. What factor is it? You have to learn. Okay, then this is the last topic. Quantitative ecology. It is the calculation topic to you. Calculation. Dia macam tapi jangan risau, don't worry about it. The calculation does not like a mathematics or there is a complex equation and the question you know him, you don't know him, one. No, it's the simple calculation only. Actually, this calculation you have learned in SPM, that is density, frequency, and percentage. Like that one. And relative density, relative frequency, and also the percentage, relative coverage. So there are total 7 formulas. You have to memorize it. You have to memorize because in examination it will not give you at all. And every year, if you see passage, every year must have one question, this kind of question. Actually, this kind of question is 3 mark, bonus mark to you. 1 mark. Your objective question only 1 mark. But also it will come out in structural question. We don't know. Maybe it will come out in structural question, or you come out in structural question, you already have 7 marks or 8 marks already. It have come out in 2 years. I think 2 years structural question have come out this kind of question, quantitative ecology. Wow. If come out, you are untung lah. Keuntungan awal kerana 7 markah sudah ada di tangan lah. If you have memorized all the formula and use the formula correctly. If you don't memorize at all, then this mark you rugi lah. Awal rugi lah. Kerana MPN sudah bagi markah percuma kepada kamu, tapi kamu tak nak, MPN pun tak boleh cakap apa, saya pun tak boleh cakap apa. So you have to memorize it. Not too much lah. 7 formulas. Matematik 20-30 formula boleh memorize, boleh sudah hafal. Biology 7 formula tak boleh hafal. Tak mungkin kan. So you have to memorize. Okay, this is the sampling method. And the second one is pattern of distribution. This is new to you. This is the population, that some of the population, if you see that some live they live individually, some live randomly, some live clump, they clump together. So total there are 3 types of this pattern distribution you have to learn. And this one is gentle reminder, that is in Oxford, there is information not too much for this topic. You have to find the information from others' book, like Campbell. Campbell have the detailed information about it, because in essay there are 1 year essay question already come out. Want to use to explain the pattern of distribution. I think 6 marks or 8 marks, like that. So you have to know. So if you just look the Oxford books are not enough to you, you have to see the Campbell books. So this chapter is finished. So in conclusion, this chapter also not too hard, and mostly have many things that already you have learned in SPM. This you have already learned in SPM. Energy flow actually you have already learned in SPM. Also this sampling method you have already learned in SPM. Others are new to you, but also not too hard, easier you to understand only. Chapter 3 is the selection and speciation topic. This is the last topic of ecology. Okay, before we enter the selection and speciation, this topic, you will refresh back that the variation. This is already also you learn in SPM. Importance of variation, continuous variation, and discontinuous variation. If you know, ingat balik. awak tahu lah apa saya cakap. If you already forget at all, please you can refer to your SPM books textbook or other SPM reference book. Variation same saja, sama awak belajar dalam SPM. No, there is nothing much to not too much thing berubah. Okay, so mainly more teacher will skip this chapter lah. They will tell you read sendiri, baca sendiri lah, kerana awak sudah belajar lah. Then you will proceed to the topic of this chapter, that is natural selection, and also other is artificial selection. It is the selection. Selection has two types: natural selection and artificial selection. So before that, you have to know what is the selection. Definition of the selection. You have to know that. And if for natural selection, what is the natural selection? Natural selection is that semula jadi punya. Environment dia sendiri jadikan dia. Tak, orang manusia tidak akan berubah. Hanya semua dia automatic punya. Manusia tak akan campur tangan untuk ini. So there are total 5 selection you have to learn, but these 3 are the more important compared to these 2, because these 3 have graph. You have to differentiate these 3 type of graph. There are graph you have to memorize, you have to know, and also some description because in essay it had come out 9 marks. Want you to describe the natural selection, stabilizing, disrupting, and directional selection, and also you have to sketch the graph. You have to sketch the graph. Maybe in essay question you want you to sketch the graph. You have to sketch the graph correctly. Not sketch the graph incorrectly. Or include the labeling lah. You sketch the graph in not label also will not get full marks. You have to label also, and some description you have to know, and you have to know also example of the species that beyond to this 3 selection. Okay, then another 2 natural selection is sexual selection and polymorphism. Actually, these 2 selection are theory, mostly theory. You can more understanding from reading. Campbell also can. Also, if you want to read Campbell or other ecology book also can. Oxford book also can. You can more reading. But in examination, mostly will come out these 3 selection. These 2 selection until now not come yet in exam yet, but we don't know. Maybe it will come out in future. We don't know. It is still in the exam syllabus. You have to know. And another is artificial selection. This is quite boring topic because only just you have to memorize only, understand and memorize. And memorize just macam artikel, macam baca essay saja. You have to know and memorize only. And this artificial selection until now also will not come out in exam yet. So we don't know, but I am not saying that you do not read. You cannot read this chapter, this topic. So awak pandai-pandai lah. Pandai-pandai lah. Saya my advice is awak pandai-pandai lah. Macam mana awak menguasai tajuk ini. And the last is speciation. Speciation is you have to know that what is definition of speciation. Speciation actually is the formation of new species. Macam mana species baharu boleh dijadikan. Dia ada caranya. Okay, then there are type of speciation. There are total 2 types. You have to know that 2 types of speciation and there are differences. Differences between that 2 types of speciation you have to know. And this 4 is the process that formation of the species. There are we have 4 ways to form the species, to form the new species, totally new species: isolation, genetic drift, hybridization, and identification. And the most important is these two, isolation and genetic drift, because there are more things you have to know. And essay question had come out. Isolation, there are two types of isolation. There are two types of isolation you have to know. And also genetic drift, there are also two types of genetic drift. You have to know that. And hybridization and adaptive variation, they are known, and they are just one type, and you have to know about their description. And all these formation of species, you have to give one example that the species is formed through this formation. What species? You have to know, give one example only lah. If you can memorize two example, it's good to you lah. Okay, so this chapter is not this chapter is short, but understanding need take a time. You need take a time to understand this chapter. What are these chapters say? So okay. This chapter also not too hard, too difficult, and not too much also. So this chapter you can easily handle it. Chapter 4. This is the first chapter under team genetic. And this chapter you need to take more comfort and serious to this chapter, because this chapter, the first, is the longest chapter in Sem 3 biology. Longest chapter. There are total 8 topics you have to learn. Banyak 8 topik Then second one, almost 8 topics, only 6 topics, only 2 topics actually you have learned in SPM only. You have only learned SPM only. That is Mendel inheritance and mutation. Others all are new to you. Other 6 topics are new to you. And another one is each 8 topic. One of the 8 topics are very long. Not short. Only 2 topics are short, and others are very long. And the third one is the hardest to understanding. Very, very difficult to understand. But if you are the student that are the people that have a higher IQ, higher thinking skill, then you easier to understand. But for normal people, it's quite difficult to you understand the topic. You have to read 2 to 3 times, then you can understand this topic. What is this topic say? So this is a gentle reminder, gentle warning want to you to make a more comfort to this chapter. So some school teacher will begin this chapter first. I think some teacher will begin this chapter first, because this chapter is too long. It need use maybe 2 months to handle. Some school teacher need 2 months to cover this chapter. So you have to focus it very well, carefully. The first is hand inheritance, that is Mendel inheritance. SPM you already learned, and in STPM you also have to learn. Mendel, who is Mendel? I think everyone is know, right? Who is Mendel? Siapa Mendel tu? Mendel, he is a person. He is the father of the modern genetic in the world, right? And it found the inheritance law. So it has created the 2 law, Mendel first law and Mendel second law. And it's monohybrid inheritance and dihybrid inheritance. Another base is law of segregation, I think. Law of segregation and law of independent assortment, another name lah. But same je, sama je. No, you have to know all this is refresh SPM you have learned only. Just refresh only. No new things. So what is the SPM that you learn? That is Mendel experiment. The first one, Mendel experiment. What is the purpose of Mendel experiment? Experiment dia guna plant apa? Tumbuhan apa? Pea plant, right? It use pea plant to make an experiment, right? Then there is two kind of experiment. Monohybrid, it use monohybrid inheritance is one characteristic only. Then dihybrid has two characteristics, right? And finally you will give the ratio. If the one characteristic give 3:1 ratio, and this one will give 3:9:3:1 ratio, right? And also before that you have to know the terms of the genetic. Include earlier. What is earlier? Phenotype. What is phenotype? What is genotype? Then what is test clause? There are test clause. And there are also some of the test clause. Also back clause. Also have reciprocal force. This almost already you already learned in SPM. Just refresh back only. Refresh only. So this chapter, this topic, mainly teacher will not focus at all because you have already learned in SPM. Maybe it will just refresh briefly only. Maybe some teacher but not all teacher. But if your teacher is good, maybe it will explain one by one to you to see your teacher pattern. And also the schematic diagram. You have to learn how to draw the schematic diagram. Also will come out one schematic diagram. Remember, like this, right? Then this is a P1 undergo meiosis. Then it will give you gamete. Then undergo fertilization, you will give you F1 first generation, like this, right? Macam ni lah. Semua awak kena refresh back balik. So if you enter this chapter, you already all the SPM this thing you already forget. Hopefully, please refresh back to your SPM books. Please refresh back. If your teacher, you know that, if you in SEM 1, SEM 2, you already know your teacher pattern, right? If your teacher pattern is like, already learned SPM, so I skip lah. If your teacher pattern like that, please, you refresh yourself, eat yourself, make a revision yourself. So, but if your teacher is teach you nicely one by one, not say that you don't want to refresh. You also need to refresh, but not too hard lah. Compared to the skip teacher one, you are more lucky lah. For those that teacher already skip, so you have to get more comfort on this chapter, because if you do not handle this Mendel experiment well, later you enter this chapter, non-Mendelian inheritance, you will make a confused between these two. Confused. Totally, you will confuse. 100%. If you not learn well in Mendel, 100%. Even though you learn well, you also maybe have a confuse between the Mendel and the non-Mendelian. So in STPM syllabus, we have 8 non-Mendelian inheritance. From this topic, you already know name of the topic. You know that Mendel inheritance that do not follow the Mendel's law. Totally not follow the Mendel law. If you learn that Mendel have many laws, it has said that one of the law is say that one of the rules is say that one gene contain 2 allele only. Contain 2 allele only. Mendel cakap punya. Macam TT taller, the characteristic of height. T is tall, capital T is tall, small t is short, like that. And another Mendel say that 2 earliest, one earlier is dominant and one earlier is recessive. Yang Mendel cakap punya. And another law is say that one gene only control one characteristic only. One gene only control one characteristic only. Yang dia cakap punya. Mendel cakap punya. Semua dia cakap ni. Tapi, but, is it true? Yes, it is true. But, is it can apply in all organisms in the world? This is a question. So, later, after the few years, until now, the scientists, further scientists already make a research that not all of the organisms follow Mendel. So, they already classified non-Mendelian inheritance. So, in STPM, we have we have 9 non-Mendelian inheritance you have to learn. Total 9. Complete codominance, codominance, multiple earlier, lactate gene, polygene, link gene, sex link gene, epitaxis, and pedigree analysis. Wah, 2 more. Banyak Banyak, memang banyak. And why I say that, you will make a confuse between these 2 topics. It will totally make you confused because this non-Mendelian inheritance actually is modified from the Mendel experiment. Totally modified from Mendel experiment. Just result of this reject Mendel only. That do not reject Mendel or say that do not follow Mendel. Do not follow Mendel. So why we say that it is non-Mendelian inheritance. So, all of this you have to know how to draw the schematic diagram. For the schematic diagram, you have to know that incomplete dominance, codominance, you have to know how to draw the schematic diagram, and also link gene and sex link gene. You have to know how to draw the schematic diagram. Others, you don't need to draw a schematic diagram. Okay, for incomplete dominance, complete dominance, that is the question. If I say that, Mendel say that T, if the gene is in hybrid form, like this, T, capital T and small t, it will give you dominant characteristic, right? That is Tor, right? In Mendel cakap punya. But incomplete dominance reject this. Means that if the gene is hybrid, then it will give you another trait. Totally, it will not give you dominant trait also and recessive trait. Another trait. So there are one example species dragonfly. Snapdragon plant. That is not follow Mendel. It will have 3 traits. One is I already forget the symbol, so I use A like A untuk A replace and like this. This is dominant recessive, and this is not called dominant or recessive, we call as hybrid. If you have hybrid like this, it will give you another trait. This is an incomplete dominance dia cakap punya. Already reject Mendel. Okay. Why we say that we reject Mendel? Later you will try to draw a schematic diagram. You will know the ratio. The ratio is different from the monohybrid inheritance. Totally different. And okay, this one I do not say too much. Then later your teacher will teach you. Another is codominance. Codominance, actually I already say. Mendel say that one gene is controlled by 2 alleles. And that 2 allele, 1 allele is dominant, 1 allele must be recessive. But in the world, in such cases, there are 2 dominant. How? Dominant allele. Or both of the allele are dominant, and they are no recessive. 0 recessive. No recessive allele. So already total reject Mendel dia cakap punya lah. Already langsung total reject lah. And another is multiple alleles. Multiple alleles say that multiple allele is Mendel cakap apa? Dia cakap satu gene hanya 2 allele sahaja. Tapi in the such cases, there are more than 2. Maybe have 3 allele. There are maybe 4 allele. We don't know. Such cases. And there are example. You have to know the example. Which example is obey codominance example is obey multiple alleles. Okay, another one is lacteal gene. Lacteal gene, Mendel cakap apa? Mendel cakap kalau if like this, dominant. If like this, also dominant. If like this, recessive, right? But there are some organism cases if they give this gene in the earlier, like if the organism have this kind of gene, it will die. Oh, mati lah. Langsung mati lah. Dia tak akan hidup. Ini boleh hidup. Tapi dia tak ada recessive trait. But dia akan mati lah. Terus mati saja. Ini total reject Mendel lah. Mendel tak cakap dia boleh survive. Just only give recessive trait. But the theory say that it will totally die at all. Cannot survive. So it also reject the Mendel. Another is polygene. Polygene, Mendel cakap apa? Mendel cakap satu gene control satu karakteristik saja. Tapi actually most organism do not have like that. Especially the complex organism like human. Actually the height of the human is more than 3 genes or 4 genes. I think there are 3 genes. 3 genes control one characteristic. This means that there are more than 1 gene lah. More than satu gene will control the characteristic of the organism. Like a human, the height of the human actually have the 3 genes control. Ini tiga gene ni akan control height of the human, not only 1 gene. So ini total reject Mendel lah. Langsung reject Mendel lah. And another one is link gene and sex link gene. These two are actually same. Link gene, if you know Mendel say that one chromosome, that is 1 gene lah. 1 chromosome only has 1 locus only. Ini Mendel. But link gene say that maybe one chromosome actually have 2 locus. Siapa cakap betul ah? Mendel cakap satu gene hanya ada 1 locus saja. Tapi ada scientist cakap, no. Actually, 1 chromosome have at least 2 locus or 3 locus. Siapa cakap betul? Semua cakap betul. Semua betul. This is just that a special case. Special case. So this is the case that you may confuse between dihybrid inheritance and link gene, because both of these include 2 characteristics. The difference are, one say that chromosome have 1. This say that 1 chromosome have 2. So totally different lah. But the same is they are involve 2 characteristics. So your teacher will tell your will sketch the schematic diagram, one of the schematic diagram for spatial for link gene. Then you will know that the result of the ratio, the phenotypic ratio of the link gene is totally different with the dihybrid inheritance. You will get the result 3-1, I think 3-1. But Mendel second law you get 9-3-3-1, right? But the link gene you get the 3-1 result. Totally different. So you have to know how to sketch the schematic diagram, and you have to know that the difference between the dihybrid inheritance in link gene, because normally the exam question will give you a question. Exam question will not mention this is the link gene or dihybrid. It will not state that, and it will ask you, is this hybrid? Is this breeding follow Mendel? Explain your answer. Dia keluar macam ni. So you have to according to that question and it will give you the result. 3991 or 31. You have to know that. But it will not give you like that lah. Like this, like general like this lah. It will give you the result lah. Result macam dia kakak ada 500 red flower, 450 white flower like that. It will give you like this. Then you have to sendiri modify the ratio. Sama ada dia ikut Mendel atau tak ikut. Then you have to expand that kind of question. So you have to differentiate well between these two. If you not differentiate well, then sorry lah, you will lose mark at all. This one teacher, I think your teacher will perlahan-perlahan teach you perlahan-perlahan, teach you nicely and make you understand. If you don't understand, please ask your teacher. Okay, so link gene and sex link gene are said. Just sex link gene includes sex chromosome only. Another one is epitasis. Epitasis is same like a polygene. Macam sama dengan polygene sahaja, tapi tak sama. There are some difference. What difference are there? You know. And P degree analysis is the last, the non-Mendel inheritance chapter. So, in conclusion, this topic is quite difficult, especially if you enter this link gene and sex link gene. It's more difficult at you to understanding. So you have to take more reading and also take notes. Make own notes to understand this topic. What are the topic talk about? And if you don't understand, please ask your teacher immediately. Okay. So these are 17.1 and 17.2. And now you enter 17.3 and 17.4. Genetic mapping. Genetic mapping actually is about the sex link, breeding between sex link. Not sex link, link gene, between link gene. But mostly teacher, if they already touch this chapter, already touch this topic, it will directly teach you lah. It will directly teach you lah, the link gene. So actually, the breeding you have learned it. You need to learn in genetic mapping, but mostly teacher will already teach you in 17.2. And another one, 17.4 is population genetic. This is calculation topic. Soalan pengiraan. Hardy-Weinberg equation. Tapi don't worry. The equation, you only learn 2 equation only. P plus Q equal to 1. P square plus 2PQ plus Q square equal to 1. Only these 2 equation. Easier. I think you can memorize, right? P plus Q equal to 1. P square plus 2PQ plus Q square equal to 1. Memorize only. I think pun tak perlu memorize lah. Walaupun boleh tahu lah. But just we have to know that what is P, what is Q, and also P square, 2PQ, and Q square. This calculation topic I cannot talk too much. You have to answer the question. I think your teacher will give you exercise. If you do exercise, then you will know. Also the bonus mark. Calculation topic is the bonus mark to you. Easier to you. So if you already know the equation, then if you use the equation well and use the sub the value, correct value in the equation, then you will get the correct answer. Okay, now you will go to the DNA replication and begin this topic. Actually, it is under genetic control lah, not under inheritance lah. Inheritance topic is finished already. So you will enter genetic control. And the first genetic control topic is DNA replication. Okay, you have to know that DNA macam mana dia replicate. Actually, if you SPM, if you know if it's SPM or STPM lah, sem 2, it have see the word replication kan, DNA replication. Tapi you don't know how the DNA actually is replicate. Betul tak? Maybe if you are the higher thinking student, maybe you already searched the YouTube about the DNA replication. But if you know not that, you will learn about it. Now you will learn about it, DNA replication. Before we learn about the DNA replication, we have to learn about the history of DNA. Sejarah DNA dia. Actually, also experiment to prove DNA as genetic material. So this is the one of the history that in early genetic studying, we don't know. Actually, we don't know that DNA act as the genetic material. We don't know. In the earlier studying in genetic, we only know that nucleus carry genetic material. You only know nucleus only. Tapi awak tahu kan, nucleus ada banyak benda. Ada kromosom, it has chromosome, it has protein, it has carbohydrate. And even though in chromosome there are DNA, DNA, and also has histone protein, right? There are many components. There are many biomolecules. So which molecule that act as genetic material, scientists don't know that. Earlier, for earlier study, scientists do not know. Some say chromosome, or some say DNA, some say protein, some say carbohydrate, but there is no evidence to prove that the DNA is a genetic material. There is no any evidence. So there are total 5 scientists that make 3 experiments. 5 different scientists in different years, different decades. So there are total 3 experiments. You have to know that to prove that the DNA is a genetic material. So first experiment, you have you have to learn you want to learn. The first thing is experiment name. Name of the experiment. Name of the experiment is according to the scientist name. So you have to memorize the name of the scientist. Name of the scientist you have to memorize. The second one is procedure. Procedure of the experiment. Dia macam mana how they carry out the experiment. Third one is result. What is the result of the experiment, and the final conclusion? What conclusion they make? You have to know all about it, and you have to know why you have to make 3 experiment. One experiment enough mah? Kenapa ada tiga eksperimen? You have to know that it actually has a reason, because the first and second experiment are not the evidence are not strong to prove that the DNA is a genetic material. There are some scientists reject that experiment. Only the final experiment, there are 2 scientists called Hershey and Chase. Hershey and Chase experiment, dia kedua-dua saintis ini telah menjalankan eksperimen mereka dan prove that DNA is confirmed as a genetic material. Until now, why we say that DNA is genetic material? Mereka kedua-dua saintis ini, kalau tak ada saintis tersebut, maka kita until now we don't know that the DNA is a genetic material. So we have to apologize to these 5 scientists. But I thought your heart is I hate these 5 scientists lah. Because of you lah, I need to learn this thing. I know your mind. I am also the STPM student. I know your mind, but no matter, you have to know that. You have to know that. Okay, so then you already finished learning the DNA history. Then you will go to the model of DNA replication. Okay, next story, macam cerita lah. Next story is, okay, you already know DNA is genetic material, but how DNA replicate? Macam mana DNA replicate? Caranya dia replicate mana? Not this ini ialah cara detail lah. Tapi before we know that Java DNA, because that time technology is not too hard, the technology is not too modern that we can see the DNA replication mechanism. That time technology not too modern like now. So we just only can guess macam mana DNA dia replicate. So ada satu scientist lagi. Actually this scientist actually you have already learned in sem 1. Watson and Crick. This scientist. Why sem 1 you learn DNA? The shape of the structure of DNA. Structure of DNA dia yang bagi kan, scientist ini yang bagi structure, prove the structure of DNA, right? So propose the structure of DNA. He also proposed the model of DNA replication also. At that time he had give three kind of model. Dia bagi 3 bentuk model. Tapi bentuk yang mana betul dia tak tahu. He just make a hypothesis only. It will give 3 hypothesis. 3 assumption macam 3 assumption sahaja. 3 hypothesis sahaja. Tapi mana which hypothesis is correct? Nobody know. Dia pun tak tahu. Actually dia pun tak tahu. It just gives 3 hypotheses. So there are another experiment, 4th experiment in this chapter. Another experiment. There are 2 scientists. I see who are they? I already forget, but you have to memorize it because experiment name is based on the scientist name. So same as this experiment, you have to know the scientist name. And the second one is experiment procedure. Macam mana the way they carry out the experiment that time, and the result of the experiment, and the conclusion. And the final conclusion is there is only one model is true for the DNA replication. That is semi-conservative. There is only one model only. Semi-conservative model is true about the DNA replication. Others 2 are totally false, wrong. Only these are true. Now, macam mana dia dapat? From the result. From the result, you can modify from this semi-conservative model. So, dia lah. Ini 2 saintis ini, mereka buat eksperimen, jadi sudah boleh tahu DNA replication is based on the semi-conservative model. Terhadap saintis ini, kita masih belajar 3 hipotesis ini sampai sekarang. Tapi sudah ada 2 saintis ini, then we know that only semi-conservative model is true about the DNA replication. Then finally, we will learn about the DNA replication mechanism. Macam mana DNA replicate? Based on the semi-conservative model. So we know lah. So, this is the topic of DNA replication. It might quite long topic and also quite difficult to understand, especially the experiment. Experiment you have to quite difficult to understand. And also DNA replication mechanism not easy to understand. Also, there are many enzymes, especially there are many enzymes. Specific enzyme name you have to memorize. For each function enzyme dia akan ada fungsi dia lah. Fungsi dia kan, you have to know that. So these are all about DNA replication. Okay, next topic is about the gene expression. So gene expression is the macam mana gene express. Actually, if what on the other word, it is also the protein synthesis. About the protein synthesis. Macam mana? You know that cell will produce protein, right? Each cell will produce protein. But how it produce? According to the gene expression. So the firstly, also you have to know the history, gene concept, history, sejarah lah. Tapi awak tak ada. I say that you learn biology, but you learn the history of biology. Gene concept, you have to know the history of the gene concept. So there are another experiment. This is the final experiment. 5 experiment. There are total 5 experiment. Bieder and Tetten experiment. These are the 2 scientists that carry out this experiment to prove the gene concept. So same, you have to know the name. The name is Bieder and Tetten experiment. Then procedure, result, and the conclusion of the experiment. You have to know that. And this experiment already come out in STPM 2020 essay question. Why I know? Because I am the STPM 2020 candidate, so I know. So this experience, you know that experiment question will come out in exam. Before 2020, no experiment question come out. Even in structural question or obituary question, no, never come out. But in 2020, MPM cakap dia akan keluar. Keluar lah in essay. In essay nasib baik lagi. Kenapa? Kerana essay awak boleh pilih. Kalau tak tahu, awak tak boleh, awak tak perlu pilih question ini, soalan ini. Tapi if dia keluar dalam structural question, habislah. 7 markah atau 8 markah sudah bye-bye lah. Kerana awak tak tahu experiment ni. So, conclusion, you have to know all of the experiment. Mana tahu dia 2021 keluar eksperimen ni. Siapa tahu? We don't know. Siapa pun tak tahu MPM ni dia apa, fikir apa. So, please read well in this chapter, in this topic. Okay, this is just a small reminder. Okay, then you will learn about the genetic code. What is the definition of genetic code and how the genetic code function in protein synthesis. If you see the DNA like that, right? DNA macam ni dua kan, dia ada macam banyak base kan. Ini base A, C, G, U, A, B, C, G, macam mana ada banyak punya. Itulah genetic code. And actually we only 3 genetic code combined together, it will produce 1 amino acid. It will produce 1 amino acid. 3 genetic code produce 1 amino acid. 3 genetic code produce 1 amino acid. How they produce? Actually, this data you will learn about in transcription and translation. This is the briefly intro part. Before you enter in transcription and translation yet, you have to know this theory first. Later, after you know the theory, then you will enter the transcription and translation. This is the process of protein synthesis. How the protein synthesis is based on these 2 processes. Actually, these 2 processes you have see in SPM, even in STPM SEM 1 and 2, you have also see these 2 processes, but you don't know the actual process of these 2. So in SEM 3, you will learn this. So these 2 processes are quite difficult. If you want to understand, it is quite difficult. So maybe you have to make a video, or you can see the YouTube 3D modeling. You see the 3D modeling YouTube, then you can more understand that, because that is the image. You have the image of the mechanism process. Okay, so now enter 7.7, the last 2 topics, regulation of gene expression. Okay, but for this we only learn about the prokaryote only, bacteria only. Like a bacteria, eukaryote we not learn. And firstly, you will learn about the operant system. What is operant? Then you learn the operant system, the structure of the operant system. Then you have to learn mechanism of lead operant system, and also the mechanism tryptophan operant. You have to know lah. But mostly we focus on lead operant system. Mostly. Not say that tryptophan operant is not important, because they are similar, quite similar. The concept used are similar, but the mechanism are different, have a slightly different. So you have to know that. Know that the difference between these two mechanisms. And the last topic is mutation. This is the SPM topic you have learned already. Gene mutation, chromosomal mutation, change in chromosomal structure, and change in chromosomal number. There are four types and two types. Actually, you have already learned like insertion, deletion, duplication, like this. Conservation, translocation, like this. When they learn in SPM, just refresh back only. No too hard lah, not too hard because just refresh. Just another, the new thing is for this topic. Change in chromosomal number. One type is like a human, like for human, Turner syndrome, Down syndrome, Klinefelter syndrome, XO syndrome, like that. Like that syndrome, right? And another mutation is for plant mutation, special for plant only. Mainly focus on plant. There are another name, I think is autopolypodia or euploidy, I think. I already forget. But there are some mutation called plant mutation. So, okay, finally finish of this chapter. So, in conclusion, this chapter very long. It need 2 months to settle this chapter bio teacher. So this chapter you have to take more comfort lah. Not easier. It's not easier. I can say that it's not easier to understand. Also, even in memorization also. All these chapter are important. There is no such thing that is not important. All the question will come out in exam. Include the experiment. Many people say that experiment will not come out lah because never come out. But in STPM 2020, it already come out. In essay question, I think 8 marks. I think it already come out. This bidder and Teton. So you see that these 4 experiment will not come out. Who knows? Who knows? So you have to know. And also the calculation question, OBJ question, every year will come out. Even though in structural question also will maybe come out. Come out. And others also important. All are important. Just mutation lah. Maybe you say that mutation are not important because mutation until now only come out in OBJ only. Structure and essay question never come out yet. Have come out. STPM 2020 already come out for gene mutation, essay question, differences already come out. Mutation is one of the topic that is core topic also, not come out in exam. Only come out in OBJ. Structure and essay never come out, but in STPM 2020 it already come out. STPM 2020 break the record lah. Banyak record lah dia keluar. Kalau awak tengok, dia banyak keluar record lah. Chapter 5. The second chapter of genetic, the last 2 chapter of this semester. Okay, this chapter actually you also learn in SPM, that is genetic engineering. But I know many of SPM students doing SPM do not focus in this chapter, in this topic because too hard. It's harder, very hard. You say that sangat susah lah. Apalah ni. So I think many people also will not. So you no need to refresh back just because you can learn like a new things to you because in SPA you learn, you also in gene technology you also learn in this and you will learn more detail. So briefly I will say about the genetic engineering that SPM you learn. In SPM, you use the insulin as an example. Also, STPM we will use insulin, but not only insulin, also have another 2 examples also. But insulin is the most example we use lah. Okay, insulin. Then if you know that the first step is that there is the DNA of the insulin, in DNA of the insulin and also plasmid, bacteria plasmid, they will cut cut down the gene of the insulin and plasmid. Dia akan gunting, cut down the gene of this is a gene, not all gene lah, short genes sahaja, part of the gene of the insulin and plasmid. Then the second step is you will insert the insulin, cut insulin gene into the plasmid. This is the insulin, and this is the plasmid. This plasmid already have the insulin gene, and then you will combine, you will diffuse the plasmid into the bacterial cell, because it is the one of the component of the bacterial cell, right? Plasmid. Then final you clone in there. That's all in SPM. SPM just learn like this only. But in STPM, we will see very detail about all process. Sama sahaja, konsep dia sama sahaja, tapi dia akan belajar lebih dalam lah. The first is removal of gene from donor cell DNA and plasmid. Donor cell DNA, we take as example is insulin. So we have to cut down the gene of the insulin and plasmid. But how do we cut? Kami sebagai pengkaji saintis, researcher, we cannot simply cut sahaja. Syok saya kat sini, syok saya kat sini, syok saya kat sini. No, there are the ways. We have to cut. And there are one enzyme we have to use to cut the gene of the insulin and plasmid. Same enzyme. Okay, this is the first. And the second one is placing the gene into a vector. Then gene this gene you will place into the vector. Vector is plasmid. Plasmid. Okay, then you have to know why plasmid is suitable for vector. Kenapa plasmid sahaja boleh jadi vector? Actually, there are two types of vector. There is plasmid and another one is macrophage. Macrophage. I have another example. I already forget it. Apa phage punya? Bacteriophage. I think bacteriophage. I already forget, but you can refer to the textbook. So there are two vector only. Why these two vector are suitable? Why they can act as vector? Why others component cannot? Why? You will learn at here. Okay. Then third is transformation. Then you will transform this to the bacteria, into the bacterial cell. This is a process we call transformation. And the fourth one is the screening and select the transformed bacteria. When you transform, actually, if you make a genetic engineering, you not just make one only. Tak akan buat satu saja, akan awak buat billion more than 100 billion punya, few million thousand punya. Awak ada satu container sampel punya. Nah, ini semua lah bacteria plasmid that already have the insulin plasmid. But can you confirm that all of the specimen here have the of the bacteria have the insulin plasmid? Can you confirm that? You cannot confirm. Awak tak tahu kan? Awak macam mana awak tak tahu? Maybe the insulin plasmid is less, so some of the bacteria do not have the insulin plasmid. You don't know. So you have to make a screening and selection. Awak perlu membuat pemeriksaan sama ada semua bakteria ini ada insulin plasmid. If tak ada, awak kena ambil keluar. Kamu hanya mau yang bakteria yang hanya insulin plasmid sahaja. So this is the way. There are the ways, two ways. I think there are the two ways to screening and select the transformed bacteria. And finally, if you already finish, then you can do a cloning process, or we can say as amplification. Then you can produce large amount of the transformed bacteria that have insulin plasmid. Then the amount of insulin also will be largely produced. This is one of the genetic engineering. Not one of the example of the genetic engineering. There are total 3 examples in STPM you have to learn. Okay, then this is the general process of the genetic engineering. Then you have to learn the genomic library and cDNA library. These two are the topic only, but you have to find the information from the others internet or other for genetic books. Because the information in Oxford books are very little. Because in got 1 year essay question have come out this kind of question. He want you to explain the differences between genomic library and cDNA library. If you just read the Oxford book, you cannot answer this kind of question. Totally cannot. Because too little, the information too little. So you have to find the information from internet. Okay, and this chapter very important because it already come out 4 times in essay. Essay 4 times already. Include to the 2020. 2020 also come out already. Genetic engineering. It 4 times already. And genomic library, this 1 times in essay. And not only 4 times in essay, 1 times in structure. Structure question have come out 1 times. So very important, right? Very important. So you have to know. So even though you SPM, you already forgive up. Until STPM, don't want to forgive up. You have to learn back. Just like a new things. Learn like a new things. Learn about this topic, this chapter. Okay. So this chapter more about this. More focus about the genetic engineering, the process of the genetic engineering. Okay. Then finally we go to the last chapter, biotechnology. This chapter actually is new chapter for new syllabus. All syllabus does not have this chapter. So it's quite they are quite not quite difficult, but average come out in the always come out in exam every year lah. I can say that every year past year you see past year every year question got this kind of question, biotechnology. Okay, for this chapter you will see actually agriculture, human growth hormone and insulin. This is use the genetic engineering concept. One of the biotech, because genetic engineering technique is one of the biotechnology. Biotechnology, what is biotechnology? You will learn the definition of biotechnology is you use biology, how you use the biology cover in the technology. How do you use technology in biology field? Like that. Agriculture, human growth hormone, and insulin is what is you will use the genetic engineering technique to this. So there are 3 examples. Agriculture, human growth hormone, and insulin. Mostly will do these 2 lah. Agriculture and insulin mostly come out. Human growth hormone never come out yet, but same saja, sama dengan insulin saja, just tukar nama saja. Okay, then the first one, food and average production. This one boring topic never come out yet. Never come out. Food include I think milk, cheese and yogurt. I think these 2 food you have to know macam mana nak prepare milk macam mana keluar punya cheese and yogurt. Beverage pula adalah beer and wine. Ini 2. Vinegar I already forget got or not, but in these 2 I know. But vinegar I already forget. If yes or yes, if no then you don't need to know. These 2. Then you have to know the process, all the process actually you have to know, but until now there is no question in past year, so I don't know how to say it. Awak pandai-pandai lah. I only say that you can pandai-pandai lah. And then another, these 3 topic you have to know detail lah. essay question always come up. Gene therapy actually is the treatment. It's a treatment process. Treatment process. DNA fingerprinting is the you can detect the information of the individual based on the DNA of the DNA, such as the hair of the person, fingerprint, or others hair or mucus of the person. Then you will detect that extract the DNA. Then we will know that the sample of the things is belong to who. Normally it is held in the criminal process, criminal process, and also relationship. This why if you have see for Chinese if you have see the drama Chinese Hokkien drama 8 o'clock punya awak tahu kan dia akan banyak DNA saya nak check DNA kepada dengan ibu dia, awak nak check sama ada ibu awak ada is the own mother or not, then you can use the DNA fingerprinting technique. Then you will show that actually your mother is not your mother. Your mother is not your mother. Maybe I don't know. This is the drama If you see the drama, you will see the story like that. Actually, it can do one DNA fingerprinting relationship. And normally we focus on criminal process. And another genetic screening is the detection of genetic disease during fetus or pregnancy for human disease. There are two types. I think there are two types. This is the one of the technology that you can detect. You can check whether your son your baby during pregnancy already can check whether your baby have genetic disease or not. Like Down syndrome, Turner syndrome, that is all are the genetic disease. You cannot make a prevention. If you got, then you have got. If you get this disease, then you will get this disease until lifespan. Your whole lifespan. There is no any treatment that treat the disease. So we call as genetic disease. We can use nowadays we can use the genetic screening to detect the genetic disease. But in Malaysia, rarely use this kind of technology because Malaysia punya mind macam can say that macam ada dahulu mind punya tak ada not open mind like negara barat lagi. So Malaysia do not use this detection. But you have to know there is already one of technology can detect the genetic disease. And another one is biosensor. Biosensor, this one actually you have learned in SEM 1, enzyme topic. SEM 1, your last enzyme topic application punya ada biosensor, right? Just same. Just you can refer to your SEM 1 textbook. And the last topic is oil decomposing bacteria. This is the one of the bacteria that can decompose the oil. It is helpful to the environment, especially the sea. Okay, there are total 9 applications in biotechnology in STPM syllabus. So in conclusion, this chapter generally come out in essay question because it is a new topic. So MPM will selalu keluar topik ini. So you have to make a comfort attention also in this chapter. So that's all today for this video. So thanks for watching. If I will say sorry about it if my English is not good or my information maybe have flaw. Now I'm saying sorry about it. You can refer to your textbook or ask your teacher for further information. And good luck for your SEM 3 examination. And if you have that repeat U1 and U2, good luck also to get your best result. And another reminder that is SEM 3 do not have repeat. So please do the best in your SEM 3 examination. Thanks for watching. Bye.